Okay. Okay. I just want to go back over this a little bit at the very tail end. We didn't. There's this new gene editing technique called Crispr. I'm not going to tell you what it means. It's a long, jargony sort of thing. But it was first discovered in bacteria. It turns out bacteria, well, everything gets parasitized if it can. And so bacteria get parasitized by viruses, and they're called phages. And they're pretty efficient. What they do, like all viruses, is they insert their DNA into the bacteria. The bacteria then makes more phages, viruses. So bacteria and archaea actually have a way, they have an adaptive immune system. And it was discovered, I don't know, fairly recently. And it recognizes viral RNA or DNA, whichever it is. And then it's got these enzymes. First, it's got this thing where it has a record of all the different kinds of viruses that have, in terms of DNA sequences, that the cells have been infected with. All the way back, from parents to offspring, all that kind of stuff. And then they've got enzymes that cut those viruses up into little pieces. Well, someone realized that you could actually hijack the whole system and use it to edit genes at very precise places. So that little part where it actually recognizes, in fact, let's take a look. Okay, so this is this is the guide RNA. In other words, you can make it anything you want. The bacteria, it's a copy of the different viral sequences. And there's different guide regions for different kinds of viruses. Well, we can actually do the same thing, but instead of using viral DNA, we just put our own DNA in there in terms of what we want the system to recognize. It then goes exactly to the genome. So here's our genome. And it goes to that sequence in our genome, the exact base pairs. And then it cuts it right at that point. And then at that point, you can either do a number of different things. You can take out some of the genes, so you knock it out, and then do experiments with mice that have the knockout and see what happens. You can also do something called RNA interference. You don't have to know about that. Or you can put in new base pairs, a new gene. Okay, so it's you've got this donor DNA in this case that you decided to put in, and you can then put it into targeted genome editing into anything you want, whether it's a zebrafish, a human cell, a bacterial cell, whatever. So, and then of course you can take that cell that you've done and you can get it to start to divide and form an embryo, and then you can have a whole line, let's say, of mice expressing that new trait, or what you hope is a new trait. Okay, so it is it's a very, very new sequence, new technology, and there's actually a big patent fight going on now. It's a little bit, well, you can imagine it can be used for all sorts of things. You can put in new genes like this. You can find out what genes do by knocking them out. You could also create transgenic animals like you do with what we learned before, but this system is much more efficient and precise. So before, with the recombinant DNA, you have to do thousands and thousands, and hopefully you get a few that actually worked. Well, with this, you can be absolutely precise about where you want to cut into the genome. And then, of course, I would imagine eventually there'll be some nefarious individuals who will take a human cell, let's say, insert certain genes that have been, they've been identified involved in certain things, let's say spatial intelligence or whatever, put those genes in and create a transgenic human being. Now, of course, it'll be outlawed, but there's always going to be someone like that. So it's kind of a scary sort of thing. Back to the end of this, let's say you've made a transgenic plant, and this, let's say it's corn, and it's resistant to all sorts of pests, insect pests. So now you don't have to spray it with an insecticide to keep those insects away. The company that made it can obviously charge lots of money for it because farmers won't have to use insecticide anymore. And then you grow it. And it's basically a clone of one type that has this new feature being resistant to insect pests, right? When you have a clone, that means there's no genetic diversity whatsoever. And let's say a new disease comes along. I'll give you an example with bananas. Anybody in here like bananas? Okay. There's actually many, many, many varieties of bananas. The ones that we generally eat are Cavendish. There's a whole bunch of other ones. Some are used for cooking. A lot of them actually are much more tasty than a Cavendish, but they just don't keep. Okay, so anyway, the Cavendish was developed, and that's all the bananas that we normally find in the store. Well, there's a new disease out there, and Cavendishes, they're all clones of each other, essentially. They're all susceptible to this new virus. And it's wiping out, and they're afraid, actually, that the modern Cavendish banana, because it has no genetic diversity, it's going to get completely wiped out. So every time the virus gets spread into a new plantation, all the trees die. It's a mess. So they're trying to now, of course, generate a new variety that's got the same sort of bland taste, and it keeps well. Okay, so in terms of these plants, you've got no diversity whatsoever. So obviously, if some pathogen or parasite comes along, it's very highly susceptible to it. And selection can't work on it, right? Selection only works when there's heritable genetic diversity. If there's no diversity among all the plants, then there's no evolution. So you can see, let's say, what happens. On the left, you've got healthy corn, gets hit with a blight, and you end up with nothing for a crop. So GMOs are certainly susceptible to that. a lot of people are freaked out about GMOs. It's really no different than what you would call artificial selection, except that instead of selecting for traits and breeding over and over many generations, you can actually insert certain genes that are then expressed.